All right, hi everybody. Hope you had a nice lunch. Mine barely touched the sides, to be honest. Okay, so let's dig out. What we're going to use. Okay. All right, so these are the data from the linkage fly experiment, fly report. I forget all the different names that I call it. Linkage fly project, there you go. That's from this table here. Right, and so essentially, what I'm going to do today is do exactly what you could get from watching the video here. This is Dr. Page's version of the same. Doesn't hurt to have kind of two different takes on it. And so kind of, well, first off, before I kind of go any further, does anybody have any questions about the cross that we're going to be analyzing? Give you a chance to ask questions rather than me just chatting away. So just as a recap, and again, kind of using that notation that you saw earlier in the cis linkage stuff and the PowerPoint. Again, what's at the top above that line is one chromosome. What's on the bottom underneath is the second chromosome of that pair. Yeah, so this is about, Dipper, two things. Pardon me. One, well, so the whole project is kind of essentially you would be doing the cross yourselves, but unfortunately we're not doing this in person. But what we're going to go through is the actual cross that you would do. The data that you would collect, we're going to give to you yourselves, but you're still going to do all of the analysis in terms of forming the graph, doing the recombination distance, so on and so forth. And then you're going to write about all of that in a mini lab report. Right? Probably be about 3 pages long, give or take. That's kind of what they work out at. And so what we're going to talk about today is how you can analyze these data, right? The different ways in which you can present them, which is important, and kind of dive into them to figure out, you know, what's going on, right? And so this is in part, last lab we talked more about the structure of the cross, like how you can get these kind of data, how you can do this kind of experiment. Essentially, we're going to work on the assumption that you've collected the data, right? And now we're going to talk about, well, what do you do with that, right? What do you do with that or those data, and how can you analyze them, process them in a way that other people can interact with and understand, right? That's kind of the key, right? That's a very important scientific skill to develop. We are writing this in a very specific format. So, actually, it's in here. Aren't I clever? So if you scroll all the way down here, and this is, which tab? Get out of it. Hang on, let me just get rid of this for a second. Here we go. Pardon me. So all of this stuff you will find in ta-da, here. Right, so this is the document that I'm working off of at the moment, this linkage fly project. And these two documents here will give you, in different degrees, information on how to write the lab report. So we will go over this again next lab, right? So the lab today is really about the analysis, right? How you can generate a graph and stuff like that. I don't really care too much about the format you put your references in. If you have references you want to cite, then stick them in there, put the references at the end, and you're good. You don't have to cite references if you, because that's not part of the grading, and it's not super important at this point. It's a skill you'll pick up later. But if you wish to, then as long as you cite them appropriately, then I'm happy with that. So anyway, all this information you'll find is here. And also, all of that should be in down here at the end of that linkage fly project document. Okay, but we're not going to talk about that much this lab because we'll have time to do that on Tuesday. Actually, when are the deadlines? Doesn't hurt to talk about that stuff. All right, so the graph and stuff which you'll see in that video, if you want to go through that too, which is a really good one, will be due by midnight next Tuesday. Right, so the Excel spreadsheet assignment due by midnight. Oh, what's going on with my typing? Tuesday, 6th of July. And that's what we're going to talk about today. The mini lab report is due, oh, hang on, scratch that. Check my calendar. All right, that's 29th. There you go, that's it. My apologies. Got the wrong Tuesday. What day was that again? 29th? Yes. It's the mini lab report. Report is due by Tuesday, 6th July, midnight, by email. Actually, that would help to put that in. And ideally, put your name in the file name. Makes my life so much easier. I don't have to rename all of your different stuff so that I know whose is whose. Okay, so those deadlines, always good to know. Where were we at? Okay, so this is the cross that we're doing. We're crossing a wild type female with a mutant male. These two genes, white and yellow, are X-linked. And you can tell that just by looking, because there's a big capital Y here. So that tells you that this is the X chromosome at the top, but this is a Y chromosome underneath. Right? Cross these two together, you'll get heterozygous females. What linkage phase are these females? A little bit of quizzing you from stuff today. Yay, they're in cis linkage. Good stuff, Maria. And you get wild type males. These ones we virtually kill. In real life, we actually kill them, but because this is an online, see, virtually. Anyway, probably not the time for jokes. So we then cross these heterozygous, it's actually called the fly morgue. It's a little container full of, I think it's ethanol. You just put them in there and they die happy, or they just never wake up, basically. So yeah, there's like little cross-eyed flies on them. So we cross those heterozygous F1 offspring with basically recessive males, again, mutant males. And that should give us 4 possible combinations. So this is exactly like the cross which we had in here. This one. The only difference here is that this is translinkage, right? So exactly the same idea. So we have 4 potential combinations. We have our parentals, right? This is what the mother looked like. This, in a sense, is what the father looked like. We're going to collect data for both sexes, right? And so we have different ways in which we can analyze these data depending on, you know, how we handle it. And we have 2 recombinants. So the result of recombination between these 2 loci in the female parent, right? And so if these 2 genes are linked, we would expect these to be present at a much lower frequency than the parentals, right? It's kind of what we learned about this morning. Okay, got any questions about that? So hopefully it makes a bit more sense this time around than it did last week, because we've actually gone over this stuff in class. Okay. So the data that we've got are basically a whole bunch of student data that we've pooled together into one large clump. And that's generated this, right? So here we have the 4 different phenotype combinations. This is going to be the mutant, double wild type, and then the 2 recombinants. So we know these are recombinants 2 ways. One, we know the phenotypes of the parents, right? Which is wild type, or mutant. Have you spilled something, Robert? I was going to warn you against shaking that thing so vigorously. Put an eye out. Second is these are present at a much, much lower frequency. Okay, so quick question. Do you, just looking at these data, would you guess that these are closely linked or loosely linked? What do you think? Just looking at it, kind of first pass guess. Yeah, closely linked, because the numbers are really, really low. Okay, so now we have two tasks essentially ahead of us, right? You can't put data in this format into a paper. It just, well, visually makes no sense, right? So one of our jobs as a scientist is to take these data and put them in a format which is a lot more visually accessible, right? And this is not a trivial skill, right? This is an important skill to learn, right? And, you know, as I now temporarily at least have a PhD student working with me, I'm going through all of this with her, right? Because she's just finished her first year of a PhD, right? So this is something that you learn. It's an important learned skill, and that's why we have it in this lab, because it will help you for later on. So we have to do that. So it's kind of like a distillation. I always think of it. You take a whole bunch of data and you kind of distill it down into something that is visually accessible. Like, for example, these are the data that my student generated for me a week or so ago. So we have a genotype. We're knocking down 3 different genes, one of which is control, and we're counting how many cells survive when you do so, right? So these are the raw data, right? These are the counts that she generated. And if you know what you're looking at, which I do because I've worked on this a bunch, you can look at that and go, ooh, that looks kind of neat. But anybody who doesn't, wouldn't make any heads nor tails of that, right? And so then you do a whole bunch of prettifying, and you end up with a graph that looks like this. And this will be the graph that goes into the paper that we're going to resubmit to the journal, hopefully, fingers crossed, published by the end of the summer. If I can kind of G up my PhD student to finishing the data collection in time. Right, so this is the end result. So you look at this, you're like, oh, wow, okay, so this one is clearly a lot lower. Whether that's better or worse kind of depends on what the axis is. And this one is a bit lower, but, you know, not as low and actually kind of closer to this one. So that's visually accessible. Even if I didn't tell you anything about this, you could tell me exactly that back. This one's really different. This one's a bit different, maybe. We'll have to do the stats from this one, whatever this is, right? So that's the first step of the job. Second step is, well, what does that all mean, right? What do these data tell us? And so that partly depends on what's your hypothesis. So there are a bunch of different statistical analyses that one can do, and we'll go through those. But those are based on assumptions and predictions. So you have to square those away first before you do any statistics. The statistics you do, or the statistical analyses that you do, are only as good as like the intellectual rigor that supports them from behind. Right? You can just kind of like, you heard the phrase, there's lies, damn lies, and statistics, right? So you can use statistics to show all kinds of funky stuff that, you know, isn't actually true. So you have to be very careful about what your hypotheses are. So before we do anything, there are 2 different hypotheses one could work with for this particular experiment. What would those be? We're dealing with linkage here. So what are the 2 extremes when you're dealing with linkage, or genes that may possibly be linked? What's the 2 extremes on either side? On one hand, they could be. On the other hand, they could be. What are those could be's? All right, so 1 Good, Rose? These two genes are, let's not even just say closely, completely linked, right? There is no recombination occurring between them at all, right? So kind of put your hand over the screen and don't look at those data, right? Because obviously you can't generate a hypothesis after the fact. It should be beforehand. So that's one extreme. These are completely linked. What's the other extreme? They could either be, obviously, most likely somewhere in the middle, but what's the other extreme from being completely linked, no recombination at all? Yeah, good job, Marissa. Not linked at all, right? So those are two different hypotheses, right? And so you can choose whichever you wish. Generally, you work on the assumption that nothing interesting is happening, right? And so typically the hypothesis that one would choose by default, if there are no other information to suggest otherwise, is that these two genes are not linked, right? So we start with our hypothesis. Right, and a big hint is that your hypothesis is the central foundation on which your entire project is carried out. So that has to be in your introduction of your lab report. Just as the hypothesis or hypotheses, because you're doing lots of different things, in that paper that I'm writing in the moment, you have to talk about it in the introduction. Even if we later find out that it was wrong, which we did, you still have to talk about it in the introduction. Okay, and so that kind of sets the stage, right? So start with a hypothesis, and I don't know, the default is that, oh, I don't know. I mean, I'm always a little bit tricky. Each hypothesis, each experiment or project is generated from a hypothesis. And I don't think there's any way to say there's a default hypothesis. I don't think that's actually correct. So, pick your hypothesis. And I want to stress, like really, really strongly, there's no such thing as a wrong hypothesis. A hypothesis is a thought construct. It's like, kind of stroking beard or chin. If you don't have a beard, you can get like fake beards, to put on so you can like stroke them while you're generating hypotheses. Or you could even do without it, maybe. I mean, I managed for a while before I had a beard. Obviously, the science I'm doing now is way better because I have a beard to stroke. But anyway, so, there's no such thing as a bad hypothesis. Hypotheses are generated by, mine's coming from Amazon. What, the hypothesis? I didn't know you could buy those on Amazon. Do I get rewards? Or a beard? That's fine as well. Sorry, it's like you either laugh or cry, right? That's kind of, you either like zonked out or you're manic, one or the other. So typically hypotheses are generated from some kind of information that you may have about the question at hand, the topic at hand, right? So, information would be, well, these genes are both on the X chromosome. I don't have any evidence to suggest that they're linked. That's why I'm doing the experiment. So my hypothesis is that they are unlinked. That's fine. You could use the obverse. Actually, you'd probably verge on that one because, there are lots of genes on chromosomes. No guarantee that they're going to be right next to each other. So let's go with that one. And usually I like being really direct about it. I hypothesize, thought I'd know how to write this better by now. that white and yellow are not linked. You could also pick that they are completely linked. Doesn't actually matter because you're going to do your analyses based on that hypothesis, right? So from that hypothesis comes the prediction that I would see an equal number of parentals and recombinants. So a hypothesis is a thought construct that generates the predictions that you're going to test. That's essentially the scientific method. Then you do the experiment and then you see whether your predictions are correct. If they were, your hypothesis was correct. If they're not, then your hypothesis is wrong. And so you try again, try a different hypothesis. Right, and so the reason why I'm banging on about this is because you have to have this before you do any statistics, because at the very least in this project, your statistics rely on those predictions. So if you're doing a chi-squared test, you have your observed data and you have your expected data, right? How would you calculate your expected data? Well, that's based on your predictions. So you have to get the hypothesis straight first. Otherwise, the rest of it just isn't going to work. And so anyway, once we've, done all that, then we can do our analyses, right? And so there's a bunch of different ways we can analyze these data, just in terms of categories, right? So we can see whether or not our observed data match our expected data. So first thing is a chi-squared test. Because that's how we figure out whether or not our prediction was accurate or not, as the case may be. Right. Second, I shouldn't say thing, analysis. There you go. And so an odds ratio is, it's actually kind of a funky thing. I never encountered one before I started teaching this lab back in 2018, I think it was, because I never used one in my own research, right? I don't do that kind of work. Essentially, that's like, what are the odds that you'll find these two alleles together, right? So what are the odds that you'll find a dominant white and a dominant body color allele together, right? If everything's equal, the odds of finding any particular combination of alleles together is going to be the same. So it's going to be 1. There's no more likelihood that you'll find these two alleles together rather than these two because, your hypothesis is that they're not linked. Does that make sense? That's kind of how I understand an odds ratio. I think that's actually accurate too. And then the last thing you need to do is, well, I can work out not just whether they're likely to be linked or not. I can actually work out the physical distance between them, or the map distance. Again, map distance and physical distance aren't 100% related, but for our intents and purposes, they're close enough to be one and the same. And so you can do the same calculation that I've been showing you today. Just take all your recombinants and you divide them by the total number of progeny. And that will give you your map distance. So really, in many ways, what you're doing today is the actual results part of a project. You've collected the data, right? That's the trudge work. Now you get to see whether or not, well, what have you got, right? So let's get back to drawing a graph. What would you expect what would you expect to be based on your hypothesis? So, good question, actually, Rose. Yes to both. It depends on how you're doing the analysis. So if you're, you could do parentals versus recombinants. I personally wouldn't do that so much because you lose quite a lot of information if you condense things down too far. So you could do a 1-to-1 ratio of parentals to recombinants. So your expected would be half of the total would be parentals, half of them would be recombinants, right? And then you add these up, these would be all of your parentals, these would be all of your recombinants. I think that would miss a little bit too much information though. So you could do it as, like you said, a 1-to-1-to-1 ratio. Doesn't matter, Rose, those are exactly the same things. So if you have 4 different options, each one of them will be a quarter, right? It's the exact same thing. So you could do it by phenotype combination, right? You could do a 1-to-1-to-1 ratio, or essentially you'd expect of your total, 25% to be the dominant, 25% to be the recessive, and then 25% each or a quarter each of the 2 different recombinants. Or you've got 2 different sexes here. Is there like some kind of sex-dependent weirdness going on? So you can do it as 8 categories, right? So could be 1-to-1, not ideal, as you lose a lot of information. So I can't actually see all of the text in the chat box because it's kind of halfway off my screen, because otherwise I can't see the rest of it. That's why some of those typos come in. Another option would be 1-to-1-to-1-to-1. Or, as you say, quarter to quarter to quarter to quarter. It's essentially exactly the same thing. And another option still would be, let's see if I can copy and paste this, because this will get real tedious to type out. Hey. Oh yeah, I did put enough in there. Right, so where now you have 8 different categories, 4 for males and 4 for the females. And I don't have any preference between the second and, oh, come on, and third. As long as you're clear about the analysis you're doing and the reason why you're using those expected values, doesn't matter. So there's actually, and this is another important point to make, there's actually a lot of leeway as to how you present this information. It really depends on what story are you telling, right? This is something that I try and stress to my students, is that there's very rarely a right and a wrong way to present data. I mean, obviously there are some really not good ways of doing it, but it really depends on what story you're trying to tell. So for example, let's Heebie-deebie-doo. All right, so we could just select all these data and go to insert graph and do that. That's perfectly legitimate. We've got our count data. We actually have to add, so you go up here, add chart element. What would that be? Hey, that worked. And then we need to have, you always have to have access titles, because otherwise what the hell are you presenting, right? No one can know. Okay, so this is a little bit of a kludgy looking graph, but it will do for now. Right, and again, pardon me, all of this is kind of covered as well by the video that Dr. Page did. So that's perfectly fine, right? You could just do that. That's one option. All right, you could also, and again, I don't want to force you into doing things a particular way. You might say, well, you're the professor, you can get to do whatever you like, which is true. But I want you to think about this process, right? Because again, what story are you trying to tell? If you care about the any kind of sex shenanigans going on, these are important things to plot out. If you're not, they're not, right? So let's say you don't care about sex or flies, at least. Let's just look at the phenotypes, right? So that there be a sum. And so equals brings up the function box over here, which has got, what is that? I2 in it at the moment. And then you select which ones you want. Now, if you want to be like real particular about it, you could just do the same for each one. But I'm not, and I'm lazy. So I just hit that little square at the bottom right-hand corner and drag it. It does it for you automatically. Now, better not lose my voice. That would be important. I have to do a whole lecture by sign language. So that's another way of looking at it. Right now, so now we've, and again, probably better do this. Right. Gets a little tedious because you get the point, but I'm such a pedant that I can't not do it. Come on. All right, or number of flies or whatever, right? It's not super important. Okay, so that's just, again, raw data. And you can take that even further. Actually, let's just type up new ones. And you can have parentals versus recombinants. And I got to tell you, I can't actually remember how Dr. Page did it. So don't feel like you're getting conflicting information. I'm just trying to, he does it one particular way. That's fine, works well. I'm showing all the different ways that you can do it. And so let's get out of it. These two. Now, unfortunately, you can't do the draggy thing because Excel is not smart enough to figure out what you actually want to do. So you have to do a second one. Now, these are pretty ugly charts, and I wouldn't leave them like this because I'm super particular about my graphs. Not everybody is, but I like mine to look a particular way. What would that be? Yeah, it's called phenotype. Uh-oh, what happened there? I've lost a bloody axis. There you go. Let's get that back. Control Z or Control Z is like one of the most useful things ever in the history of Office software, as far as I'm concerned. So you can also just click on that as well. Count Dracula. Anyway, so those, I know, I'm sorry, kind of scraping the bottom of the barrel for jokes right now. It's tough. My comedic abilities have decreased. So these are just, essentially, they're just different ways of presenting the same data, right? Depending on what you want to say. And each person has their own particular proclivity depending on what it is they're saying. You can do more though, right? So let's take this and we've got to copy and paste this as values. Otherwise it will try and calculate sums from over there and that would be weird and wacky, right? What about what we expect? So let's sum these all up. Oh, that's what it is. So this is that times 0.25. There you go. Can kind of work out in my head, but in some ways I'm kind of lazy. So right, those are our expected values, right, based on our original hypothesis. And so now we're not just presenting the data in a form, we're actually doing like an analysis of our data by saying, well, these are what we observe, but how does that fit what we expect? Obviously, we do that formally using a chi-squared test, because that's like the proper way of doing it. But we can do that visually too by actually plotting that on a graph. Where are we at? Go. And so now you can see we've got a much more visually accessible piece of information. Come on. Obviously, it looks like a dog's breakfast, right? Because we haven't done anything nice to prettify it. But the information is all there. Let me bring up the chat, make sure no one's been. Oh, oh boy, I've been doing this for years. And you know, the funny thing was I was talking with my former postdoc supervisor and this PhD student about how to draw graphs. And they use this piece of software called Prism, which I've never heard of, but by all accounts, it's pretty good. And I said, yeah, that's probably way better than Excel because Excel is a bit of a dog's breakfast when it comes to drawing graphs. It kind of makes you do stuff you don't want to do. But for what I do, it's perfectly fine. And so I said, you know, I've been doing this for 15, 20 years this way. I'll just keep doing it the same way. It works for me. So it's not that I'm any kind of whiz. It's just I've been doing it this way, low, you know, over and over and over and over again. You just kind of get used to it. And you can do the same for any of these, right? Obviously, there's a kind of a balance, right? And it's kind of scientific, kind of aesthetic too. Pardon me. Between putting the information on here and putting too much information on, such that it becomes difficult to actually really access what's going on. So if you, a good example would be, if you add the observed and expected to this chart, which you could do, it'd be a bit more kind of finagling around to get that to work the way you want it to, but you could. So, but then you'd have, what would that be, 16 columns. And so it's possible now you're starting to hit the limit of, well, you actually make things less understandable by putting more information on than more understandable. Same could be said for over here. Here. Oh, yeah. If I was Kiwi, that would be what I'd be saying. Oh, yeah. Bucket of prawns. Sorry, I watched part of, what is it? Yeah, that's it. Avengers Endgame last night. And that guy that's a collection of rocks, he's got a Kiwi accent, which whenever I see it, always makes me go, oh, yeah, fish and chips. Anyway, so flip side is now we're starting to lose information, right? Now we just have parentals versus recombinants. So that's a subjective call that you get to make when you draw these graphs or whatever graphical visual way you wish to use to represent your data. There's not really any right or wrong. Obviously, like doing like starburst charts or whatever that I can't understand, that's not great because the aim is to make this stuff accessible, not have lots of like flashing lights and stuff. So I'm going to leave this sort of up to you, right? How you want to do this. Obviously, if you watch the video that Dr. Page makes, then he has his own particular way of doing it. He'll show you that way. And that's totally fine as well. You're not going to get dinged. There'll be no like 0 marks. You didn't do it like the favorite way of your professor or whatever, right? The aim is to just show you that, well, there's actually some degree of art involved in this process. At what point do you start, you can kind of make it more and more accessible, but at what point do you start making it inaccessible through what you do? And then obviously you don't want to leave just chart title up here. I don't know, you can put whatever like you want up. You could do that. And be like, super amazing, absolutely awful. I don't know, I'm just being silly now. So, I don't know, I actually never put chart titles, to be honest, because that's what the figure legend is for. So let's can that. There we go. Better. I hate lines on graphs. This is one thing I'm like super particular about. So typically if you right click on anything in Excel, as long as you're right click in the right place, it'll bring up a, let's change this. So let's format grid lines. Space. I wish I had a bigger laptop sometimes. All right, let's get no line. That looks better. Somehow, I don't know why, we don't have a sorry, I'm gonna have to move you over here, Robert. You're still there. You're just not gonna be covering up the bit of my Excel sheet. There we go. Let's have a nice solid line. But whenever you do that, for a reason, this is the flip side of Excel, Adam. It is a solid pain in the butt when You try and do something it doesn't do automatically. There we go. You can make the font bigger. You can do mostly whatever you like. I don't like borders around graphs. For some reason, it always looks weird. But again, that's just me. No border. And you can fiddle far around with this as as much as you want. You can change colors. If you click on one and then you click on it again. Oops. Come on, get out of it. Right now you just have that one. You can pick a particular color. Oh, blue, blue. Go red. Oh, that was the outline. There you go. Let's do the fill instead. Because for some reason, Excel always gives you like these kind of semi-lame ass colors. I don't know why. You can do like fancy fill. Well, I would if it let me. Come on. Oh, it's over here. There we go. That's why. Don't want a solid border. We want red. Red. And again, you don't have to do any of it like this. You do whatever you like, really. As long as it does, as long as it makes kind of sense. Oh yeah, that's the border. I want fill. Let's do pattern fill. Pattern fills are always fun. If you're a Pink Floyd fan, you can do bricks. That's a bit of an old reference. Most people probably won't get that. Doesn't matter, right? I probably wouldn't do that because bricks look kind of weird in a graph. But anyway, you can fiddle far around this to your heart's desire to get it looking how you want to look it to look. And essentially that's graph drawing. So it is a skill. It's not a super hard skill to learn. It just takes a bit of practice. Hence, that's why we're doing it. And it kind of makes a difference, right? You can go from having something that kind of looks like that, which doesn't look terrible, to something that looks like that, which looks fabulous. And you can drag it around, make it bigger. You can make the actual chart area bigger. Click on it in the right place. Like so, you can do whatever you like, really. Anyway, any questions? Extra special befuddlements. Will you hate us for using lines? No, I won't. If you like using lines, that's totally fine. Again, like this is, yeah, everybody has their own like peculiarities, and lines in graphs is mine. Hate them. Don't know why. It's totally random, like no particular reason to it. I can actually see the argument of why they would be useful. I just don't like the aesthetics of them. I think they look like crap, basically. Not that I'm judging you. I mean, if you think they look beautiful, that's totally fine by me. Again, that's the art part of it. Yeah, totally. I mean, that's a perfectly valid reason for doing it that way. There's better reason for me not doing it that way, if you see what I mean. Mine's just me being peculiar. Yeah. Whatever you like in terms of aesthetics is totally fine. It's a totally subjective deal that I have no beef with. As long as it doesn't get in my way of actually accessing that information. You could have little prancing unicorns filling the. Oh, there you go. While we were talking about unicorns, you can have a pretty pony off in the corner if you want, as long as it doesn't get in the way of the graph. Awesome. Maybe I should start giving like extra credit out for like unicorns or something. If you could find a fill which is unicorns, that would be pretty cool. So that's graph making, straightforward, easy peasy. Chi-squared is also pretty straightforward. You just have to be clear with yourself. What am I analyzing? How am I doing the analysis? So Dr. Page has given it to you in terms of each of these pieces of data are individual categories, right? So if that's the case, you have 8 different categories. You have your total divided by 8ths to give you your expected. So if you scored, yeah, that works. If you scored 1,000, your expected for each of these would be 125. And so you do the chi-squared. So these, let's say these are your observed data. And so if you look real carefully, 5 through 8 are the males. 1 through 4 are females. And these are the data that you collected, right? So each of these are treated as their own separate categories. If your hypothesis is that there's no linkage, no recombination will be happening in every round of meiosis, you would expect 1/8 of these for each category, 1/8 of the total for each category, right? That would be your expected values. Then you just carry out your chi-squared accordingly, right? You would do category 1 would be 243 minus 125 squared divided by 125 plus 229 minus 125 squared divided by 125. And you do that for each of these, right? That's kind of what's laid out here. That would give you your chi-squared value. Then you work out your degrees of freedom. And then you go back to Blackboard and work out your p-value using those 2 values. Or you could do it where you don't care about sex at all, right? So you group these together like I did here. And now you have 4 categories, right? So now your expected values are going to be 1/4 of the total. So it'd be 500, 500, 500 in this case, or in the case of this, it would be this example down here, it would be 250, 250, 250, right? Because you would add the males or the females, sorry, and the males for each category together. That would be your observed. Your expected would be 250. Then your degrees of freedom is 3 because you have 4 different categories. So it depends on how you want to do it. There's no wrong way, but you just have to be straight in your head, like, what am I testing? So you do the experiment properly, or the analysis, really. Like, you wouldn't do chi-square with 8 and then go, well, I've got 4 different categories, and you'd have degrees of freedom of 3. That would be wrong, right? It would give you an inaccurate result. Then there's the odds ratio, and that's super, super easy, right? Essentially, all it is these 2 times together divided by these 2 times together. That gives you a really big number. And that's basically the odds of you finding 2 alleles together any given time. So that's a very, very high odds. I'm not a betting man, but I would bet on that because that would be like pretty much guaranteed. So that's the odds ratio, and it's really well explained here. I'm not going to go into it in a great deal. It's simply a matter of making a table like this, doing the calculation here, plug in the value in. And then recombination, again, is pretty straightforward because all you're doing is you're adding up your recombinant offspring divided by the total. That will give you your recombination frequency. Now, just to be really, really, really clear, the examples that I've used here in these different things are not necessarily the data that you would be, or the results you would be getting, because you would be doing all of those with these data here. Okay, just to be really clear. And that's not just to prevent you from copying and pasting stuff, although that's kind of a little bit of it, is to make you go through the actual process yourself. Again, Dr. Page does a great job of taking you through that too. And then that's it for the Excel spreadsheet assignment. So what I want from you is basically an Excel spreadsheet with a graph, sheet 1 or sheet 2, doesn't matter. The other sheet, the analysis, you could actually even do it on the same sheet. It doesn't really matter, as long as I can find everything. Of the chi-squared, odds ratio, and the recombination distance. Hand that in, do all of those, 100%, easy peasy. And the plus is that when you write your mini lab report, you're going to take all of that, dunk, put it in your mini lab report. You're not going to have to do it again. So you've already done the preliminary work for your lab report. Then you just have to kind of fill in the gaps. So for the lab report, that's going to be the result section is the big one, because that's the important one, really. All the things that you do for the Excel spreadsheet assignment, the graph, chi-squared, odds ratio, recombination frequency, plus, people often forget this, a bit of descriptive text. Tell me what you found. Don't just rely on those pictures and those numbers, actually tell me, right? And I think there's a figure legend in there somewhere as well. But we can go over that when we talk about the lab report. Cool. Anybody have any questions?
So do you only want that descriptive text for the table when we do our lab report, or do you want it for this assignment as well? Okay.
No, for the lab report. So this assignment is really getting you to prepare all the things that you're going to talk about in your lab report in terms of results. But they're just like the bare analyses. You're not going to talk about them for this one. That's all going to go into the lab report, which is due on the 6th. Yeah.
All right, gotcha.
And really it's, you know, in a way I've made it a little more complicated than it needs to be, showing you all these different things. It's really quite straightforward. And also, if you follow like what Dr. Page does, you'll get everything that you need. Not sure if he does the odds ratio in that video or not. Yeah, but either way, that's what you need. And that's pretty much it. So let me just write that down for posterity's sake. Get a bloody chat box up again. So one Excel file, obviously. In that should be a graph with axis titles at least. Need a chi-squared analysis. Odds ratio and map distance in centimorgans. That's centimorgans, not centimeters. All by midnight. What did I say again? 29th? Yeah. And that's pretty much it. Anybody got any questions? If you do, and you don't think of them between now and whenever I end this, email me them or bring them to lab next Tuesday and we'll have time to talk about them there. I'll still give you time to finish the assignment before the deadline, midnight of that day. All right, cool beans, everybody. Thanks for holding in there. I shall see you Tuesday. Remember what day of the week it is. Have a great weekend, everybody.